Rapidly optimizing upstream processes and seamlessly scaling up continue to be top industry priorities. The Gibco Efficient-Pro system provides a next-generation CHO medium and feed system to help address these goals by offering an enriched medium and feeds to support performance improvements in CHO-K1, CHO-K1 GS, CHO-S, and DG44 cells. I'm Miles Skoda. This is the Life Science Connect webinar series, and today we're talking a next-generation medium and feed system to improve productivity in CHO cell lines. Now, joining me for this presentation are two industry experts from Thermo Fisher Scientific. First, Dr. Anna Hochman, Senior Manager for Research and Development, and Ryan Boniface, Global Product Manager, who together are going to detail results from the use of this chemically defined offering designed with an innovative multi-omics-based approach. Now, we'll be holding a Q&A session following this presentation, so please feel free to submit questions as we go along. We'll get to as many as possible during the Q&A, and any that we don't get to, we'll follow up on after the event. And then just a bit of housekeeping: if you run into any issues today, please try refreshing your browser first. If your problem persists, submit a question through that Q&A, and I'll help get you back on track. And then finally, this event will be available on demand, usually within a day, often less, and you'll get an email with a link to it, or you can just use the same link that you used today. So with all that said, I'm going to turn it over to Ryan to get started.
Thanks, Miles. So what we'll be covering today are some industry challenges, highlights of the Efficient-Pro medium and feed system, product productivity, quality, and performance data, a scale-up case study that we performed with Zurich University of Applied Sciences, what's on the horizon, and then we'll toss it over to Q&A. CHO cells remain the predominant cell type with our industry. They continue to evolve to produce ever higher titers. And we're always looking to provide new products that are easy to use, that can support higher titers and cell densities, reduce development time, have consistent product quality. That means not just our product quality from lot to lot, but also the customer's product quality, meaning the product quality that they're getting for their titers and their protein, and of course, scalability. So with that, I'd like to introduce the Gibco Efficient-Pro medium and feed system. Efficient-Pro offers versatility, meaning we can support multiple cell types and proven to produce mAbs and complex proteins. Performance, enriched medium and feeds to support high viable cell densities and titer. And then scalability. We offer both liquid and AGT formats backed with security of supply. Highlights of the Efficient-Pro platform include chemically defined AOF and multiple formats, including AGT and liquid. Feeds designed for all variants of CHO cell types. Higher titers with maintaining product quality. Ease of use with our AGT format, and up to double the protein production when compared to Dynamis. So to touch on ease of use, as mentioned, Efficient-Pro is offered as an AGT. This eliminates the need to do multiple pHing steps and mixing steps, resulting in dramatically reduced reconstitution times. Ultimately, you're just adding the AGT to water, you're mixing, and then filtering, and then you're ready to use the product. I'd like to now turn it over to my colleague, Anna Hochman.
Thank you, Ryan. In the development of the Efficient-Pro medium and Efficient-Pro feeds, we utilized multi-omics technology, including proteomics and metabolomics. Much like athletes require different nutrition, whether you're a marathon runner or a weightlifter, different CHO cell lines have different nutritional needs. Ryan mentioned, we have developed different feeds for CHO-K1 and CHO-S cells. The development involved culturing the cells in bioreactors and taking samples over 14 days. The samples were analyzed with mass spectrometry for proteomics and metabolomics. And this allowed us to get data on more than 1,000 metabolites and more than 7,000 proteins, as well as how they fluctuate at different time points during the bioreactor experiment. The datasets were overlaid to identify the pathways that have the largest effect on protein titer. The development was combined with spent media analysis and design of experiment to further optimize the formulation. Once the formulation was finalized, we converted it to a dry format, AGT or advanced granulation technology, which enables faster dissolution, as well as auto pH and osmo, compared to powder media, which means it does not require supplementation of acids or bases during reconstitution. Next, we're reviewing the culture conditions. We evaluated product performance with CHO-K1, CHO-K1 GS, CHO-S, and DG44 cells. These cells are expressing IgG1 and IgG2 molecules, and they were recovered and then adapted in each basal medium for a minimum of 3 passages in shake flasks until the doubling time was consistent. Cultures were expanded, and we set up a 14-day fed batch process in triplicates for each condition in amber 15 bioreactor vessels. We measured productivity with cell growth, viability, titer, as well as specific productivity at multiple time points, as well as critical metabolites that are known to impact the cell's health. The Efficient-Pro medium and another supplier's medium were supplemented with 1% anti-clumping agent and 6 millimolar L-glutamine, except for the CHO-K1 GS cells. Our CHO-K1 GS cells required supplementation with insulin. The amber 15 conditions were set to 50% DO with an agitation of 1200 RPM. Most of our cell lines reached high cell densities, and we fed glucose up to 6 grams a liter when the concentration dropped below 3.5 grams a liter. The other suppliers' feed concentrations were based on the manufacturer's recommendations. Our Efficient-Pro Feed 1 was fed between 1 and 3% daily, and Efficient-Pro Feed 2 was fed between 1.5 and 2.5% from days 3 to 13. The results from the study showed that the Efficient-Pro medium and feeds demonstrated comparable or improved titer and higher specific productivity for the tested cell lines compared to the other suppliers' medium and feeds. In these graphs, the blue bars represent the protein production with Efficient-Pro medium and feed, while the gray bars represent the titer with another supplier's medium and feed. The lines highlighted in light blue indicate the viable cell density with the Efficient-Pro medium and feed, while the dark blue lines represent another supplier's medium and feed. In the top graph, we're looking at CHO-K1 cells that were fed with Efficient-Pro Feed 1, which resulted in protein titers that are comparable to the competitor, but promoted high viability and lower cell density, which indicates a higher specific productivity compared to the other suppliers' medium and feed process. In the bottom graph, for the CHO-S cells, the results show that the Efficient-Pro Feed 2 enhanced protein production by 44% over the other suppliers' medium and feeds. During the 14-day bioreactor run, the cell viabilities were maintained above 90%, with comparable cell densities to the other suppliers' process, which also indicates a higher specific productivity, and that can be beneficial for downstream purification. For the same cell lines, we evaluated the glycan profile of a commercially relevant IgG1 antibody. The glycans were evaluated by collecting supernatant at the end of the 14-day bioreactor run. In the top graph, we evaluated the glycosylation profiles of 3 lots of Efficient-Pro medium and feeds, a CHO-K1 and CHO-S cell line expressing a monoclonal antibody, and compared it to another supplier's medium and feed. The glycan analysis indicates the monoclonal antibody expressed in Efficient-Pro medium and feeds had an increase in less differentiated glycans over the competitor. An increase in less differentiated glycans allows for better customization to a target glycan profile. In the bottom graph, we evaluated the charge variant profile of Efficient-Pro medium and feeds with CHO-K1 and CHO-S cells and compared it to another supplier's medium and feed. The charge variants with the Efficient-Pro system had stronger neutral peaks compared to the other supplier's system, which was designed for these monoclonal antibodies. We next evaluated performance with different cell lines. In the top graph, CHO-S cells were tested in an amber 15 bioreactor. Triplicate reactors were set up to evaluate Efficient-Pro medium with Efficient-Pro Feed 2 with CHO-S cells for IgG production over 14 days. Efficient-Pro Feed 2 was fed daily at 2.5%. It demonstrated increased protein production in combination with Dynamis medium, shown in light blue, and maximized production with its complementary Efficient-Pro medium in dark blue. Therefore, we recommend pairing the feed with the Efficient-Pro basal medium for optimized results. In the bottom graph, we performed the titration of Efficient-Pro Feed 2 with DG44 cells. The feed was tested at 1.5 to 3%, and compared to a commercially available medium and feed process. The results show that the Efficient-Pro Feed 2, when supplemented at 1.5% daily, achieved optimal performance, highlighting the importance of feed titrations. During the 14-day bioreactor experiment, we monitored key metabolites. Day 14 values are summarized in this table. With the Efficient-Pro system, the ammonia levels stayed under 10 millimolar, and lactate levels stayed under 2 grams a liter for the tested cell lines, lower than the levels obtained with a competitor product. Overall, the metabolite profile observed with the Efficient-Pro system has a positive effect on cell health and protein quality. We next compared performance with other cell culture media. Here, we evaluated protein production with Efficient-Pro medium and Dynamis medium with CHO-K1 and CHO-K1 GS cell lines in combination with Efficient-Pro Feed 1 and Efficient Feed C+. Performance was improved by using Efficient-Pro Feed 1 and optimal when pairing Efficient-Pro medium with Efficient-Pro feeds for both cell lines, shown in blue. Our team has also evaluated further process improvement with the addition of animal origin-free peptones. Here, we evaluated performance with addition of either cotton or wheat peptone to Efficient-Pro medium or feed. On the left, for CHO-K1 cells, we added between 1 and 3 grams a liter of the peptones to the medium and observed up to 40% higher titers with addition of 3 grams per liter cotton and wheat peptones. For the CHO-K1 GS cells on the right, we got the best results with cotton peptone addition. We evaluated addition to the medium or to the feed and found that cotton peptone led to a 50% performance improvement when added to the medium and a 75% performance improvement when added to the feed. Of course, it is also important to evaluate effects of peptones on the glycan profile. For the same peptone supplemented conditions, we evaluated the effect on glycosylation. We observed a slight increase in G1F and a slight reduction in MAN5 with cotton peptone supplementation with CHO-K1 cells on the left. With the CHO-K1 GS cells on the right, we noticed only minor changes in glycosylation, and MAN5 stayed under 5% with addition of the cotton peptone, either to the medium or to the feed. Not shown here, but there were almost no changes to the chart variants when we tested them. Overall, we recommend monitoring the glycan profile after peptone addition. Now I would like to hand it back to Ryan to review a scale-up case study.
Thanks, Anna. I'd like to take a minute to talk about a successful scale-up study that we had with the Zurich University of Applied Sciences. What we did in this experiment is we took Efficient-Pro medium and Efficient-Pro Feed 1, tested with CHO-K1 cells, and we were able to successfully demonstrate scalability from an Amber 250 to a 3-liter high-performer bioreactor, and then all the way up to a 50-liter DynaDrive. Here you can see the results from this experiment. On the left side, you can see the viable cell densities and viabilities, and you can see that we've maintained comparable data through each one of those scale-ups. And on the right side, you can see that we maintained comparable and consistent titers from the 250 to the 3-liter all the way up to the 50-liter DynaDrive. So Efficient-Pro is an entire platform of products. It's Efficient-Pro media and 2 feeds. They were designed to work best together, but we know that our customers like flexibility, so they can be used and are compatible with other media and feeds. And then you can see on the right here that Efficient-Pro can work with a wide range of cell types. We have our recommendations here, but of course, you can try each one of our media and feeds in combination with your cell type. The Efficient-Pro system has been successfully used by multiple clients with a number of complex proteins. These clients were able to improve their titers, and they were able to do this while going from process development all the way up to GMP scale in less than 1 year. So I'm really happy to introduce the newest Efficient-Pro feed prototype. These are new feeds designed to support higher performance in CHO-K1 GS cell lines. We are currently looking for beta testers, and we'd be offering beta testers Efficient-Pro medium, a feed, and a supplement. Additionally, we will also provide a protocol, and then also support through our field application scientist team. I'd like to now hand it back over to Anna.
Thank you, Ryan, for introducing the new Efficient-Pro Feed 3. In conclusion, I want to summarize the key benefits of the Efficient-Pro system. The Efficient-Pro medium and feeds provide improved protein titers and protein quality. They're designed to provide a simple adaptation. And in addition, they support high specific and volumetric productivity. The ease of use is a key differentiator with our HT products, since no pH and osmo adjustments are necessary. The feeds are highly concentrated, simplifying the feeding process. The system provides a platform solution suitable for a range of different cell lines. Lastly, the Efficient-Pro product line provides innovative performance, and the Efficient-Pro medium and feeds are designed to work optimally with each other and have been shown to enhance each other's performance. With this, I would like to thank you for joining today, and we look forward to answering your questions.
All right, Anna and Ryan, great presentation, awesome stuff. This is really exciting, and we've got a ton of questions rolling in. Audience members, just a couple reminders on your screen. In the top right corner is the information about our speakers, and then also their contact information, LinkedIn profiles. You can get in touch with them directly if you did have some more specific questions. Below that, I've got a few resources for you. Just 3 things to download. You can click on those. It'll take you right to our site, and you can access those. And then in the bottom left, we've just got 2 quick questions for you. A brief survey that you can hopefully answer during the Q&A as we're here. And then mentioned in the Q&A at the center bottom, you can submit your questions as we get through these remaining questions. So let's start off with this first one from Jay. So for the omics part, is protein more important or the metabolites in general for CHO cells higher VCD and viability?
Can you repeat the question, please?
Oh, yeah, no problem. So this one's from Jay. If you're looking for it, it's number 9. So for the omics part, is protein more important or the metabolites in general for CHO cells higher VCD and viability?
Thank you. Yeah, good question. So for the omics part, it's really complementary because we combined the data from the metabolomics and the proteomics to optimize the medium and the feed. And therefore, we recommend doing both. If you can only do one, that's certainly helpful. But to paint a full picture, we recommend doing both. Thank you.
Perfect. Yeah, thanks, Anna. All right, so up next, Jacobo is asking, what is the concentration, grams per liter of protein under the best culture conditions and the impact over the quality of this protein?
Okay, with regard to the titers, depends on the cell line and the clone, as well as the molecule being produced, whether it's a monoclonal antibody or a different type of protein. And we have seen titer ranges between, I think, 4 and 6 grams per liter for most of our cell lines, but some other cell lines we're testing, they're getting 7 to 8 grams per liter. And we have seen concentrations up to 10 grams per liter as well with some other manufacturer cell lines. So really supports a wide range of cell lines, and the feed concentration can be adjusted for that as well.
Awesome. Thanks, Anna. Up next, so this person mentioned, they said you may have mentioned it, joined a bit late. Is the Efficient-Pro protein-free media?
Yes, it is protein-free. Yeah. Mm-hmm.
Okay, awesome. Up next from Peter, you present percentage improvements kind of on the charts we were seeing, but what titer ranges are you seeing with this new system?
Thank you. Yes, I think similar to one of the previous questions, titer ranges depend on the cell lines, so can be really between 1 gram per liter for low producing cell lines to 10 grams a liter for higher producing cell lines. supporting a wide range of titers.
Yeah, awesome. Thanks, Anna. All right, moving on to the next one here. So what are your recommendations for adapting cells to Efficient-Pro medium?
For the adaptation, okay, yes. So it depends where the cell line is coming from. If it's coming from a chemically defined protein-free medium, then it should be a very straightforward adaptation to the Efficient-Pro protein-free medium. And generally, we recommend to culture for at least 3 passages. And it's best to see that you have a consistent doubling time. That really shows that you have a sufficient adaptation. And if it is coming from a medium with protein supplements, then we recommend to add the same protein supplements, or it might take a bit longer to adapt and wean the cells off these protein supplements. And if it's coming from serum, then it might take also a bit longer. So it might be 6 passages or more, but it's certainly feasible. So yeah, we have not seen, with coming from other Gibco cell culture media, we have not seen adaptations that take really longer than 3 to 6 passages.
Okay, perfect. Thanks, Anna. Before I get moving on to some more questions here, which we'll have time for, and we've got a bunch rolling in, just a reminder, if you did have any really specific questions and wanted to reach out to our speakers, just in the top right corner, you should be able to just click on the little blue circle with the email icon in it, and that'll let you message them directly for anything else. And then, if you do have questions now, it's in the bottom center of your screen. And so let's take a second. I'm looking through the list here just to kind of organize them up. We've got quite a bit, and we're going to get back into these in just a sec. Okay, so how much insulin would you supplement for CHO-K1 GS cells?
CHO-K1 GS cells, yes. We have tested several CHO-K1 GS cell lines, and the majority of them required insulin supplementation. Some do not require that. And so depending on the cell line and the medium it's coming from, it can require up to, I would say, probably between 1 and 5 milligram per liter insulin. If you have a chance, you can titrate the amount to test it out. And if you can only test one concentration, I would probably start with 1 to 2 milligram per liter insulin.
Awesome. Thanks, Anna. All right, another one here. So what can be done to prevent lactate accumulation if the clone is prone to lactate accumulation?
Great question. So for lactate accumulation, that sometimes depends on the clone or the culture conditions. And there is multiple steps that can be taken to improve that, to reduce lactate accumulation. So one option is to work early on in the fed batch process to adjust the pH not too early. So when you start the bioreactor culture, you can let the pH naturally drop to a lower pH, about 6.8, 6.7, before actually starting pH control by adding base. That can help the cells shift their metabolism to consume lactate instead of further accumulate lactate. Another method is to improve the glucose feeding to a more tight glucose control. So oftentimes we only measure glucose once a day and then feed based on the consumption. But if possible, for a clone that is prone to high lactate amounts, you can try measure glucose twice a day and then have a tighter control to keep the glucose closer to 1 gram a liter instead of really letting it accumulate too much. Certain feeds have high amount of glucose as well, so that should be taken into account when when adjusting the feed amounts as well as when feeding glucose.
All right, good stuff. All right, up next from Solange, do you have a comparison between the 2x efficient feed and this new feed?
Thank you for the question, Solange. So for the comparison, yes. So we have shown some data in this presentation, and you can go back to the recording, of course, anytime later. But yes, we have compared it to the 2x efficient feed, especially the feed C+, and we see performance improvements compared to the efficient feeds with the Efficient-Pro feeds as well. And so some of the data we have shown is both with the Efficient-Pro feed 1 and the Efficient-Pro feed 2. Yeah.
All right. Yeah, perfect. All right. So just as a reminder, audience members, like Anna just mentioned, I do have a question. Is this available on demand? It is. If you came in late and need to watch the beginning or anything like that, it's usually just a couple hours. I'll say 24 just in case, but often much less. You can just use the same exact link you used today. It'll open it up on demand, usually shortly after we wrap up. But you know, at latest tomorrow. And so let's see what else we have here. Going through a few questions. If you do have any more questions, definitely feel free to submit them. I'm not sure we're going to be able to get to every single one today, but I pass these all along to our team here, and they will follow up with you after the event, likely via email. So let's see. From Indira, she's asking, or they're asking, suggestions for feeding days other than daily.
Yes, absolutely. That's a possibility. We have compared, for example, feeding every other day, which is certainly more convenient for some processes. That is an option as well. And sometimes it works to just double the feed concentration if you feed every other day, but it's worth it to compare different feed percentages. You can also do a continuous feed if that can be programmed into your bioreactor.
Perfect. Up next from Shashi, they're asking, is this medium specifically designed for fed batch processes, or can it also be used for perfusion?
Yes, so we have designed the feed for fed batch processes, the medium and feeds, yes. But the medium can also be used for perfusion. If you already have a perfusion process, so you can evaluate the Efficient-Pro medium, we also have a medium called high-intensity perfusion that can be evaluated as well. That was even more specifically designed for a perfusion process. But both media have been used in perfusion processes as well.
Okay, excellent. Up next from Rain, how do the feeds affect product quality attributes such as change variance and glycan profile?
So with regard to the glycosylation, we did not see much change to our standard processes. A lot of manufacturers, for example, like the protein quality they get with Dynamis with Efficient Feed C+, and we took that as a comparison, as well as media and feeds from other manufacturers, and we saw a pretty comparable profile. We saw some differences with the charge variants with our clones that were producing IgG1 and IgG2 molecules, and there we saw a slightly higher amount of the neutral peak, and that was desired for the profile for these molecules. So it's certainly worth it to evaluate the glycosylation and charge variant that you're getting with your molecule as well. But overall, we did not see significant changes comparing it with some of our existing media and feeds as well.
Okay, awesome. Let's see. Try to get a few more in here. Coming in from Colleen, they're asking, "What could you talk about the adaptation from ex vivo media to this one?"
Yes. So if you are culturing cells in ex vivo medium, it's a very straightforward adaptation to the Efficient-Pro medium. So we think we cultured them for 3 days, 3 passages, and then got a good adaptation to the Efficient-Pro medium.
All right, awesome. Thanks, Anna. Up next, so what is the feed regimen in the experiments, bolus or continuous, and is it expected to see any difference between both strategies?
Good question. So here we used mostly a bolus feed. So we added the feed percentage daily all at once. We had also compared some process optimization in the benchtop bioreactor with continuous feeding, where we actually even adjusted the feed based on the viable cell volume. And that we were able to get 10 to 20% tighter improvements with this process. So if you have the capability to adjust the feed based on how the cells are growing to really maximize your productivity, that's something we can recommend.
Okay, awesome. I'm going to just try to get one or two more in here. So let's do this one. Someone's just asking, how can we get a sample of the new feed?
Yes, Ryan, I would like to hand this question to you.
Sure, thanks, Anna. I would say that you can either reach out directly to us now that you have our email addresses that you can contact us. If you are in contact with one of our local or regional field application scientists or any of our sales folks, through any of those means, we should be able to get you samples of this product or these products.
Okay, awesome. Thanks, Ryan. We got another one just came in here from Lacan, I think. And so let me go through that one. So regarding the use of the efficient medium and feed for transient expression of protein, can you confirm if the medium and feed has been designed to avoid inhibiting chemical transfection reagents?
Oh, thank you. Yes. So it has been designed for stable production mostly, right? But when you're using chemical transfection reagents, for example, lipid-based ones like lipofectamine, then we recommend using a different medium. So there are medium components that can inhibit lipid-based transfections in the Efficient-Pro medium, and then a medium like the ex vivo expression medium can be used, or a Dynamis medium can be used as well.
Okay, awesome. Thanks, Anna. I think we're actually going to wrap up here. So audience members, in the center of your screen real quick, I do have a brief survey. If you could answer that, I'd really appreciate it. And then in addition, there's some related content as well about the topic. There are some more specific questions that we did get in that I've logged to pass along to our presenters, just for them to follow up afterwards. But with that, we want to thank you all for joining us. Anna and Ryan, thanks so much for this awesome presentation. And this is really new, exciting stuff. And so with that, we hope you all have a great rest of your day, and we'll see everybody next time.